Now let's talk about the asymmetry of lipids. As the name suggests, asymmetry means which is not symmetrical in structure, means which are not equally distributed. They do not have equal proportions of something. So lipids are also not equally distributed in both the monolayer of the plasma membrane. It means that the compositions of the outer leaflet um, and the inner leaflet are somewhat different, means they do not have the same composition and same type of lipid in both the layers. Now let's see what happened actually. So these are the lipid bilayer, and we are discussing about the asymmetry of lipid. It means the outer leaflet and the inner leaflet do not have the same amount of the phospholipid or any type of the lipid, either glycolipid or sterol. They are not uniformly present in both the outer or the inner leaflet. Some lipids are usually present in higher concentration in the outer leaflet, while some are present in higher amount in the inner leaflet. While there are some lipids also which are uniformly distributed between both the layers. So let's discuss one by one. Basically, asymmetry is one of the essential features of the lipid bilayer of the plasma membrane, because it usually essential for the normal functioning of the plasma membrane. And once it got established, then it is maintained by number of the factor, either the diffusion of the lipid structure, or with the help of the protein, as proteins always act as a transporter for various of the molecule, and lipid are also the one of them, and some protein mediated transport, means with the help of the protein they transferred from the one layer to the outer layer. Usually the motion is in the single leaflet, but there are certain movement of the lipids throughout the membrane. So these are only possible with the help of the protein. Now let's see the asymmetry means in what proportion or in what composition they are usually present in the lipid bilayer. Phospholipid, as we have already discussed, are of the four type. Phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, phosphatidylinositol, and sphingomyelin. These are the most abundant type of the phospholipid which are usually found in the plasma membrane, and out of them, the amine-containing lipid are usually present on the inner side, that is toward the cytoplasmic side of the plasma membrane, which include the phosphatidylserine, phosphatidylethanolamine, and inositol. Inside or the inner leaflet, 90 to 95% of the phosphoserine are usually present the inside the layer of the plasma membrane, that is the inner leaflet of the plasma membrane. while sphingomyelin and the phosphatidylcholine are usually present on the outer leaflet of the plasma membrane, that is toward the exoplasmic space. Okay, exoplasmic space. Now, what about the glycolipid? Glycolipid are usually present on the outer leaflet of the plasma membrane. They have the carbohydrate part, which is usually exposed in the exoplasmic side of the plasma membrane. In the glycolipid part, the carbohydrate part is usually present in the um, outer side of the plasma membrane, which helps also in some recognition of the cell-to-cell signal and some other moment-like structure. The carbohydrate play basically a major role in um, the structural and some signaling part also. Now the third one is the sterol. Cholesterol is the most abundant sterol lipid are present in the plasma membrane, and cholesterol are uniformly scattered in both the layers. The yellow part is the cholesterol one, and it is uniformly distributed in both the leaflet of the plasma membrane. Okay, see how the phospholipid and the glycolipid are usually present. Let's have a look. As we have discussed, the ethanol containing, or we can say the amine containing lipid are usually present on the inside, that is these three structure, phosphatidylcholine, sphingomyelin, and the glycolipid are usually present on the outer side. Means majority. I'm talking about the majority. Some part may also be present in the inside, but majority of them Majority of them are usually present in the exoplasmic site. Similarly, the amino-containing lipid are usually present on the inner leaflet of the plasma membrane. I will tell you the reason why it is so, because the phosphatidylserine are usually the serine containing compound, and it has a negative charge over it, and they act as a it has a tag which means eat me. Okay, so majority of the serine are usually present on the inside. If if in case the majority are present on outer leaflet of the plasma membrane, then it gives signal to the macrophages that all the macrophages should come and eat me. Then whole cell will get digested by the macrophages. Usually what happens when the cell is in the inactive, or some repair part, or some cell got disturbed, then its phosphatidylserine usually arrange themselves in the outer side of the plasma membrane. Then it gives signal to the macrophages that now I am waste, you should come and eat me. So only then the phosphatidylserines are arranged in the outside of the plasma membrane.